Good afternoon. My name is Anna Gnida, and it's a pleasure for me to conduct the second part of our Microlectures, which is entitled The Microworld at the Service of the Man. Our first among three lecturer is Klaudia Krzykała, who is a graduate of biotechnology at the Faculty of Natural Sciences of University of Silesia. Currently, she's working as a molecular biologist at Torchlight Genetics in London, focusing on innovative methods of DNA utilization in medicine. Her primary scientific interests revolve around genomics and the application of new technologies in science. Her lecture today is focused on the role of jellyfish and firefly in vaccine development. So let's listen.
Hello, welcome on the presentation prepared for the European City of Science Katowice 2024. My name is Klaudia Krzykała. I graduated biotechnology from the Faculty of Natural Science at the University of Silesia, and I currently work as a scientist at Torchlight Genetics in the cell biology group. On today's presentation, we will talk about how jellyfish and fireflies help us improve vaccines. To answer this question, we need to explain how living organisms produce light. They do it in a process known as a bioluminescence, like fireflies that emit light on the warm summer evenings as a result of the luciferase activity. Luciferase is an enzyme that combines luciferin, which is a substrate, oxygen and ATP, and produce oxyluciferin, which is the main product, and visible light, which is a side product of this reaction. Another example involves jellyfish that look colorless in the natural light, but when we illuminate them with ultraviolet light, we can see a faint green-blue fluorescence around the rim of their body. This green light is coming from a green fluorescence protein that is present inside the cells of this jellyfish. This protein is able to absorb ultraviolet light and emit green light as a result. Before we dive into the GFP and luciferase used in the vaccine research, let's follow a brief history of bioluminescence research. Already in 350 BC, Aristotle, who was a Greek philosopher, observed that fireflies produce a cold light, so a light that is cold to the touch. In 1700s, miners were using fireflies as a safe source of light. Use of open flame could cause explosions due to the accumulation of flammable gases inside the main shafts. In 1880s, Du Bois, who was a French pharmacologist, discovered that the root cause of the bioluminescence in the beetles comes from heat-stable organic molecule that he called luciferin and an enzyme that he called luciferase. Both words come from Latin luciferi, which means light bringer. In 1960s, Osamu Shimomura was able to isolate the GFP or green fluorescent protein from A. victoria jellyfish, and his discovery did not gain much traction for the next 30 years. It was not up until 1994 when people in Martin Chalfie's laboratory expressed GFP in C. elegans neurons. C. elegans is a model organism used in the biological research, and this was a huge step forward for the technology, showing the scientists that we can take a gene from a jellyfish, introduce it into these colorless warm neurons, and then be able to see exactly where those neurons are inside the warm body. And this discovery made a lot of people really excited, and then a lot of scientists started to work on this subject. And it took only a year for Roger Tsien team to create colorful proteins with increased fluorescence. And as a showcase of this technology, his team drew a sunset over the San Diego beach using only the bacteria expressing different color variants of the fluorescence proteins. GFP technology was so groundbreaking for the biological research that those three scientists received a Nobel Prize in chemistry in 2008 for their hard work. When we do basic comparison between GFP and luciferase, GFP emits light in the presence of ultraviolet light, while luciferase emits the light in the presence of luciferin, oxygen and ATP. Oxygen and ATP are already found inside living organisms, so it's enough for us to introduce luciferin, which is a non-toxic small molecule, to a cell or organism to make luciferase glow. GFP is more stable and has longer shelf life, and luciferase has bright but short-lived signal. GFP requires more complex equipment for the experiments like flow cytometers and fluorescent microscopes, while luciferase is a simple test that can be performed with basic equipment like microplate readers. There are many ways to categorize vaccines. I will focus on two general types. The first one is prophylactic vaccines, and they induce an immune response that protects against infection or disease on the subsequent exposure to the pathogen. So those are the vaccines in traditional understanding, in traditional sense. If we draw a timeline of before and after the disease, they will occupy this left side. And here we include vaccines against COVID, polio, tetanus, or influenza. The other type of vaccines are therapeutic vaccines, and they are used after the infection already occurs, and they aim to induce the immune system to alter the course of the disease. And unsurprisingly, they occupy the right side of this graph. And here we have vaccines against cancer, HIV, HPV, or diabetes type 1. What connects all those diseases is that they develop when our immune system works incorrectly. With cancer, our immune system is unable to detect and kill the cells that started to mutate and then grow without any kind of restrictions. On the other hand, in the case of diabetes type 1, our organism decides that the cells that produce insulin are the enemy and starts to kill them. And after some time, it leads to a situation where our organism is no longer able to regulate the level of sugars inside our blood. So now, if we can stimulate our immune system to find and kill cancer cells, or if we can stimulate our immune system to be more tolerant towards the cells that produce the insulin, then we will be able to alter the course of the disease. And that's the whole idea behind those therapeutic vaccines. Those vaccines are fairly new. The first cancer vaccine was approved for use in USA in 2010. In my work, I focus on the nucleic acid vaccines. So vaccines where we use DNA or mRNA. DNA is nucleic acid that is present inside the nucleus, and it contains genetic information and instructions for protein synthesis. Promoter, they are special kind of DNA sequences that signify the start of a gene. They are usually positioned in front of the gene and decides which genes are active in specific cells. Some promoters are general and they drive expression of the proteins in all cells of our body, while others promoters are tissue specific and they will allow the cells to produce brain, kidney or lung specific proteins. The next step in the protein synthesis is mRNA or messenger RNA. mRNA is nucleic acid that carry the information from the nucleus inside the cytoplasm. And cytoplasm is a place where the protein synthesis takes place. Proteins are the molecules that we are interested in from the vaccine perspective, because those proteins are used to train our immune system, and in the context of vaccines, we call them antigens. Each antigen has an optimal amount that will properly train our immune system. If we have too much of the antigen, then our immune system may overreact and cause a big inflammation. And on the other hand, if we have too little of the antigen, it will not stimulate our immune system enough to build a robust immune response. From the previous slide, we know that proteins are encoded in genes, and GFP and luciferase are special kind of genes called reporter genes. They are genes whose protein product can visually report the location of a particular protein or cell, and they allow us to monitor molecular processes in living cells and organisms. This definition may look complicated, so I will use brain bows to explain it. Brain bows are brains, for example, mouse brains, where scientists used constructs. Yes, construct always has a promoter and the reporter genes, where the promoter is specific only for the neurons. So any protein that is localized after the promoter will be expressed only in neuron cells. And then the reporter genes were different color variants of green fluorescence protein. And this means that the brain cells will express random color variants of the fluorescence proteins. And this allows us to see how neurons are built and connected with each other. In my research on vaccines, I use the cell culture and transfection techniques extensively. Cell culture is a method to grow large amount of cells inside plastic flasks or petri dishes. Cells grow in growth media that contains all required nutrients, amino acid, and vitamins. And because we can really precisely control the media ingredients, we can also really precisely control the growth conditions for those cells. On the right side, we can see a photograph of HEC293T cells, which are human embryonic kidney cancer cells. And those cells are very often used by me in my research. Those are adherent cells, which means that they attach to the bottom of the flask and they grow as a flat layer. And then to decide to test our vaccine candidates, we need to introduce those DNA or mRNA nucleic acids inside the cells, and we do it in a process known as transfection. On this photograph, we can see a result of my simple transfection experiment with the use of 293T cells. Under the normal microscope, the cells transfected with reporter gene look the same as untransfected cells. But when we look on the same cells in the GFP channel, we can clearly see which cells are transfected because they are producing this green fluorescent protein, and which cells are not transfected because they remain black on the image. And this confirms that our DNA or mRNA construct can express the gene that we are interested in. And looking on those two photographs answer the question, why do we use reporter genes in the vaccine development? As I said before, each antigen has an optimal amount that will train our immune system in the most efficient way. And that's why we need to optimize both the candidate sequence and the delivery method. If we would try to transfect the cells with our target antigen, say cancer proteins, then all the cells would look like this top left photograph, and we would not be able to easily say if the cells are transfected and if they are properly making a protein. With the reporter genes, we can optimize vaccine candidates, structure and delivery methods before we try to introduce them into the humans during the clinical trials. And this cuts a lot of time that otherwise would need to be spent on more complex techniques to find sequences, constructs that will be able to properly transfect the cells. And now we should have a general overview of what GFP and luciferase is and why they are used in the vaccine development. So let's talk through two examples of the antigens that I decided to show during today's presentation. The first example involves therapeutic DNA vaccines on the example of Listeria monocytogenes. Listeria monocytogenes is a modified bacteria into which we can introduce genes coding cancer antigens. And then after we add cancer antigens to this bacteria, it will start to present or show them on the outside of its cell. And because the antigens are visible on the outside, our immune system will think it's an actual cancer cell and will start to learn how to kill it. To date, this bacteria was used in 30 cancer vaccine clinical trials. And in the research that I decided to show, scientists selected 30 different promoters and created promoter GFP constructs. So the GFP gene was always the same. The only thing that was variable was a promoter. And then they introduced those constructs into this Listeria monocytogenes bacteria and then measured how much protein they will produce. Based on this data, they created a ranking of promoters from the strongest, which means that they produce the most protein, to the weakest. And with a data like this, scientists are able to select promoters that will be the most suitable for their vaccine application. And on this slide, we can see a microscope photograph from the experiments. It shows the promoters in order from the strongest, which was Pearl 329, to the weakest promoter, which was Pearl 31. And like we can see, this Pearl 31 promoter failed to produce any kind of fluorescence, which means that if we try to express cancer antigen under control of this promoter, we wouldn't be able to stimulate our immune system at all. The second example involves prophylactic COVID-19 vaccines. And on this slide, we can see a schematic structure of mRNA. mRNA doesn't contain the promoters because they are present only in the DNA, but they have a few other important structures. The first one is the 5' cap, which protects mRNA from degradation. On the opposite end, they have long poly-A chain, which is mostly responsible for the construct stability in the cytoplasm. In the middle of the construct, we have the gene that we are interested in, because this is a protein that will be produced. And then we have those 5' and 3' UTR sequences, which stands for untranslated region. And those sequences basically tell the cell where the protein synthesis should start and where it should end. In the experiment that I choose to show, scientists were evaluating different factors that impact vaccine efficacy and stability, and this includes different variants of those 5' and 3' sequences. Both Moderna and Pfizer created different pairs of those two sequences for use in their COVID-19 vaccines. And in the presented experiment, scientists used 5 different constructs in total. One was a control construct, where they used 5' and 3' UTR sequences that they had available in their laboratory. Then they had one pure Moderna and Pfizer construct, which were identical to those used by those companies in COVID vaccine. And then they created two constructs where they mixed Moderna with Pfizer and Pfizer with Moderna 5' and 3' UTR sequences. They loaded those constructs inside a lipid nanoparticle to mimic how the vaccines are used in humans, because in humans the mRNA constructs, they need to be loaded into the nanoparticles in order to deliver the mRNA inside the cells. And then they use those mRNA constructs loaded into lipid nanoparticles to vaccinate mice. And those photographs show results of this experiment. After scientists administered luciferin to make the luciferase glow, luciferase activity is measured in a special machine where mice are under anesthesia, so they don't move. Camera measures the light intensity and shows it as a heat map, where the brightest regions correspond to the most protein production and to the brightest signal, while the blue purplish areas correspond to the lowest luciferase activity and the protein production. And this method is so precise that it can detect just a single luciferase producing cell inside the mouse body. And using luciferase allows scientists to measure how well our vaccine candidates produce the protein without doing harm to the animals. In a summary, green fluorescence protein and luciferase are proteins isolated from A. Victoria jellyfish and beetles like fireflies. They are used as reporter genes, showing us the location of proteins and cell types, and both prophylactic and therapeutic vaccines use them during the development to optimize the antigen production and delivery methods. There are many more events and lectures planned for later in the year, so don't forget to check the information post on the Miasto Nauki website. The whole cycle was funded by the European Union and is organized by a consortium of institutions visible on this slide. And thank you for your attention.
Thank you very much, Mrs. Krzykała, for this very interesting lecture. Our next lecturer is Ravi Shankar Yadav, who is a PhD student at the Department of Environmental Biotechnology of Faculty of Energy and Environmental Engineering at Silesian University of Technology. He actively participates in numerous national and international conferences, and his scientific interest and works as well focus on biochar, ecotoxicology, bioenergy, bioremediation, and microbial electrochemical technologies. However, today's lecture, I suppose, will be more general, and its title is Microbes, Our Little Allies. So, Mr. Yadav, the stage is yours.
Dzień dobry. Good morning, everyone. So, my name is Ravi Shankar Yadav. I am a research scholar at Salesian University of Technology. And today we will talk about microbes, our tiny ally. So, this is, we will explain, like when we talk about the microbes, we always afraid of it. We thought, okay, if it is microbes, probably it will harm us. But no, actually, all the microbes are not harmful. There are very few microbes that are not friendly to us, that is pathogenic, okay, that is harmful. But most of the microbes, those are our friend, those are our tiny allies. So today we will talk about that, how they are friendly to us. So first of all, we will talk about microorganism or microbes. So any living organism that we can't see with our naked eye, we will call it microbes. So there are different kinds of microbes. Normally we can talk about, we know bacteria, that is a unicellular prokaryotic, and next one is a virus that everyone we know, that is acellular, that means it can't live, we can't say it is a living organism when they are alone, but once they infect our human life or infect any life form, then they start to act like a human organism, living organism, sorry. And then fungus, that fungi, that is also prokaryote like yeast and mold. And algae, that is also, this is a photosynthetic eukaryote. It have a ability to produce their food by themselves using photosynthetic process. And then protozoa, so that is, protozoa is also eukaryote, it is a very tiny allies. So these are a normal common microbes that we know in our daily life and in a scientific way as well. So once we will talk about the size, so we can see this is the parameter of different size. It starts from 0.1 nanometer to 1000 millimeter, that is so in this way we can see in which size range which kinds of microbes are coming. Like if it is, we will talk about 1 nanometer, the lipid are there. When it come to flu virus, it come after 10 nanometers. And then bacteria, the bacteria also follow between before 1 micrometer. And then the plant cell, frog egg, chicken egg, and then we are a human. So these are the size range, and we can also see in which size range which kind of microscope we can use to see it, to detect. So in this way, this is the different size range, that electron microscopy, light microscopy, and what we can see through our naked eye. And then we will talk about benefits of microorganism. So what is the benefits of microorganism? Because we talk about microbes are our ally, that means our friends. So how it is friends, and so how they benefit us. So there are different various way that they benefit us. So we will talk about first, so microbes in nature, how they benefit to the nature. So the different kinds of microbes are in nature. So they are, how they are working in different way. So they are a good composer, natural decomposer. That decomposer means they break down dead organic matters, like if they have any wood, leaf, any organic matters, not even only wood and leaves, if they have any dead body, that those are very smelly or something, These are the microbes. They decompose it. They decompose and make organic fertilizers. They degrade complex compound to the simplest compound that is very useful for our soil. And next, that one is a nitrogen fixer. So how nitrogen fixer? Everyone I think know about that. That they have some specific plant. So in that plant, some bacteria are there like rhizobium, azobacters. Those are the bacteria. They stay inside the plant root. They make nodules. And how they play? They do a nitrogen fixation. So how they do that? They do with them their own mechanism. They take nitrogen from the environment and they fix it into the soil. So in that soil, plant can utilize it for their own nutrition. Next one is soil formation. So as we told, so it is a decomposer. They also enhance the soil quality, as well as they also contribute to soil structure, how the soil would be more fertile, how the soil will be formed. Similarly, it also play for bioremediation. Bioremediation, that means cleanup of pollutants in the environment. So they have a different kinds of bacteria, different kinds of fungus. They degrade the pollutants from the environment, from the oil, from the soil, from the liquid water. So they degrade heavy metals. They also degrade oil, petroleum. They also degrade different kinds of pollutants in our nature. So next, it is microbes in industry. So how industrial microbes, how it work for the industry. So definitely everyone have ate bread, beer, yogurt. So these are the common beverages, normal food and beverages that we use in our daily life. So we know that those all are product after fermentation. So the fermentation is the key process to produce these kinds of food and beverages. So in this way, these all are made up by the microbes, like yeast. So we use yeast and bacteria to produce this kind of products. So the fermentation we use different kinds of microbes to make different foods and beverages. Also, because of the fermentation, we produce biofuels, that is like ethanol, biodiesels. Not only that, hydrogen, methane, these all are biofuels that we produce using fermentation. And what cause fermentation? Different kinds of microbes, like different bacteria and yeasts. Next is biotechnology. In industry, biotechnological industry, there are lots of enzyme is produced for laundry detergent, for pharmaceuticals like insulin, antibiotics, and these all. So for that process also we need bacteria, and we use like recombinant DNA technology. That means we do some genetic modification with the bacteria or genes, and then we produce different kinds of enzyme, insulins, antibiotics, and other many human beneficiary products. And for the waste management also, for wastewater treatment, the biological based wastewater treatment is the key that we produce. We treat wastewater from our domestic life, like domestic wastewater, industrial wastewater, and other wastewater as well, to reuse it and to degrade the pollutant level, COD level in our wastewater. So there is one example we would like to say, that is like a microbial electrochemical technology. So what actually it? So this is microbial fuel cell. So in microbial fuel cell, this is the like a bioreactor. In that way, there is a one anode and one cathode. So anode is normally bioanode. So normally bacteria are enriched on that anode, and those bacteria have a special feature to produce electricity using they produce electron, and that electron flow through the circuit and they produce electricity. So in that way, there is a special bacteria, those name is electroactive bacteria. So these kind of bacteria normally have a nano wire. In the picture you can see this is the nano wire like structure is attached with the bacteria, and through that they transfer electrons from one cell to the another cell. So in that way they form conductive biofilms. So that using that conductive biofilms they produce electrons. And in this way, they treat wastewater as well. They take organic pollutants, organic matters from the wastewater to feed themselves, and on the byproduct they produce electricity and electrons. So what scientists do, they change some thermodynamics and then they use these techniques in another way. Like same electron is transferring from here to there, the same bacteria are here, they close oxygen supply. In that way the oxidation process don't happen, and they produce hydrogen like photon. They using they have cathode have some catalyst. They use 2 photon H plus and produce hydrogen. Like hydrogen is also a green energy. So using the same technology, same bacteria, just change some thermodynamics and then they start to produce the hydrogen. So like this, they have a lots of different, we can use those bacteria in a very different ways to produce different kinds of products, and it very helpful for wastewater treatment and waste management as well. Another is microbes in agriculture. So how microbes is beneficial for agriculture, I think mostly people know about that. So as we talked before, the soil health. So in the soil health, the nutrition recycling, disease suppression, so they have a different kinds of bacteria. They can enhance the plant growth and nutritional quality, like because of the nitrogen fixation. Like they have so many other metabolic activities that microbes do. They also suppress some disease and like biomass fuel cells that we have already talked about, that the bioremediations. So in plant growth promoting microorganisms, there are lots of, like they have some example we can we see in the example list, like rhizobium, azobacter, mycobacterium, bacillus, azobacter. So they have lots, like pseudomonas, clostridium. So these are different kinds of bacteria that use for the plant growth and beneficial for the agriculture. So but for plant growth promotion, like symbiotic relationship, biofertilizers also. So these are different kinds of bacteria and microbes that is very beneficial for the agricultural terms that we see here. Another is microbes in medicine. Like medicine is really very important for everyone. So in medicine also, microbes are very important and they play a very crucial role to develop medicine, to synthesize more medicine like that. So first of all, we will talk about the probiotics. Here, everyone know about the probiotics benefits. It beneficial, and these are probiotics are beneficial gut bacteria that normally, like in our stomach, intestine, they have also lots of bacteria. Those are very friendly to us. because of those bacteria we can digest our food very well. We can get some extra nutritions like vitamin B and some other. So those bacteria help us to get these kind of benefits from the food also. So they also improve the digestive health. So these are the probiotics. These also contain bacteria. What are probiotics? Those are bacteria that help us to improve our digestive health, and like we talk about the gut bacteria. For another, that is antibiotics. For the production of antibiotics, we use bacteria, different kinds of bacteria to produce antibiotics. What is antibiotics? That the chemicals that are enzyme that kill the harmful bacteria or any bacteria or microbes. So that is antibiotics. To produce those kind of chemicals or enzyme, we also need bacteria. So normally we always treat infectious bacteria using antibiotics, and revolutionized medicines also, they have lots of that we use bacteria to produce for the production of those medicines. Vaccines, like recently we know the COVID-19, it was a recent example of the vaccines. So for the many vital vaccines, for the production of those, we need microbes. So we need microbes and bacteria to produce those vaccines. Okay. Next is microbes in biotechnology. It is a similar like for genetic engineering. In biotechnology, the main feature is gene modification. Gene modification using recombinant DNA technology. In that technology, bacteria, yeast and other microbes they play a very crucial roles. So in that way, like bacteria is very important for genetic engineering also, for creating genetically modified organisms, like for some plants also, for animals also, and other also. It is also advancement in agriculture and medicines, as we talk. An industrial enzyme using the biotechnology, there are lots of industrial enzyme like biodegradable plastics. It also using bacteria we can achieve these kind of targets and goals. And biofuel production, we have already talked about this before. So next one is environmental cleanup, that is bioremediations. So here we can see for the bioremediations, they have different kinds of bacteria and microbes. They play a very crucial role actually. So like we talked about before, the wastewater treatment, polluted water, and what we do, we use bacterial remediations, fungal remediations, microalgal remediations. And using these kind of microbes, what we get treated water. Okay, so these are different kinds of use for environmental cleanup also. Like they have a lots of bacteria, they can eat oil, spill oil when we go to the when they have a sea, and there are lots of shipments are going. Sometimes some accidents happen, and in that way those incidents are using like that accident what they do, for that incident, what happened, they spill some water like petroleums and diesels, crude oils into the sea. So they have lots of bacteria they have invented, and they use those bacteria for their food, and they clean the sea. So they are very crucial roles. They also can do clean up and break down the hazardous chemicals. There are lots of chemicals like cyanides also. They have so many bacteria that can eat cyanides, and also many chemicals that is very hazardous for the human health. And similarly, mycoremediation, if we use fungus for the remediation, for the cleanup, and that is called mycoremediations. For the removal of heavy metals from the soil also we use fungus. So these all processes are like bioremediations, microremediations, also some microalgal remediations as well. So in microalgals, they use photosynthesis and they produce so many good things and they clean the pollutants. Next is microbes in space exploration. This is a recent publications I found on from some scientific community group. So for the exploration, this is a very interesting topic. They use bioengineered microbes that can produce different kinds of taste. So using 3D technology, they produce bacteria or microbes, and they can form, using 3D technology, they can make burger, and we modify that bacteria as a burger taste, and then they can enjoy that burger in the space also. So this is a way, in Nature Communication, this publication has been recently published, and that support when our space like astronauts go to the space, it is very their food, they have very their foods are too much concerning things, because they can't grow the agriculture there, so they have a fruit problem is always there. How much pizza they will bring there, how much burger they can bring there, they have a space limit also. So in this way, this is just alternative the scientists start to find. So the bacteria, they have a very high protein content, so they can do some gene modification in that bacteria, and they can add some taste and color and flavor enhancing genes into the bacteria. And then what scientists can do, they can bring this bacteria, they can grow this bacteria to the space. Of course, they have a setup, and then using 3D printing, they can produce whatever they want with the similar taste. Okay, so these are the interesting inventions by the scientists recently. And microbes in art and culture, we talk about the fermented food. So in many fermented food, using the bacteria, they produce different kinds of textures and different kinds of color. So in microbes normally, color and textures, we can see in vinegar, when we produce wine, beer, cheese, they all have different kinds of texture and color, and those make food more looks delicious and make more attractable, we can say. So these are the use of microbes in our daily life, but still they have lots of challenges and ethical considerations. So what kind of challenges we know? Definitely, as we talk about, there are most of the bacteria are friendly to us, but still there are so many harmful bacteria that we know in our environment. Those are called pathogenic, infectious. So we always have to regulate, safety and regulations we have to consider. So for the contaminant of harmful microbes, so we also have to be sure that there is no any harmful bacteria contaminate myself and our environment. That is very lethal for us. So these kind of concern, ethical consideration, we have to always take care of that. So ethical consideration in genetic engineering also. So once we do the genetic engineering, if they have some other gene manipulation we do, we don't know what the impact would be in the future. So for that genetic engineering, genetic modification, we have to be concerned sometimes. So that in that way, the ethical consideration is required. Okay. And another is resistance and overuse. If we will use if we will use too much antibiotics, what will happen? Our bacteria will get resist to that. When they will understand, okay, this kind of chemicals, antibiotics they are using to us. So what they will do, they will develop a defense mechanism that can counter that in kind of antibiotics. Normally, doctors also don't suggest too much antibiotics to us, because once bacteria will know about that antibiotics, they are very sharp. What they will do, they will create some mechanism in their own self, and that can defend the bacteria, that kind of antibiotics. So in future, the same antibiotics won't work in the same bacteria. So environmental impact of industrial microbes use. So these are the ethical issues and challenges that we have to always take care of that. So at the end, we are going to conclude the all things. So recap of microbes benefits as we talk. It benefits everywhere: industrial, environmental, medicinal, in space exploration, art, and the culture as well. And so this is how we can say the microbes as our tiny ally. Their role in shaping our world for the better. It is not a destroyer. So what we have to do, we have to take care of them very patiently, but we also have to be careful for the some various infectious and harmful bacteria as well. So this is our responsibility, the importance of responsible use and conservation of the bacteria that we have to respect. Thank you so much for everyone, and if you have any question, please you can write. Thank you. Have a good day.
Thank you, Mr. Yadav, for making us aware of the beneficial role of microorganisms. Our third and the last lecturer is Dr. Szymon Gorgoń, who originates from University of Silesia, where he was primarily interested in oogenesis and developmental biology in annelids. Then he was working on designing of novel gene inhibitors concerned with certain types of cancer. Currently, Mr. Gorgoń is working at University Paris-Saint-Clair in France, where he studies proteostasis in the germline of nematode. In our last lecture, he will tell us about Cenarabditis elegans, the lord of the non-ringed worms. Mr. Gorgoń, the stage is yours.
Hello everyone. First of all, I want to say thank you for the invitation here today and the opportunity to talk to you about my work. So today I will tell you a little bit about nematodes Caenorhabditis elegans, short C. elegans, we call it also in lab worms, and why so many scientists are interested in this species, why it's present in so many labs, and why there are meetings across the world where people meet to talk just about C. elegans. So first of all, we have to answer ourselves what the model organism is. It is organism used to study some specific biological phenomena, hoping that this phenomena and this discovery from one species can also answer to the question how this phenomena works in other species. And here, when we say other species, we think mainly about humans. So this model organism have to be very simple to make it easy to dissect a given process. Also, it is important to have a right species for the right questions. So we start with problem and question, and based on this, we use the model. For example, if we want to study problem solving, we would use this spider, Portia. This small spider looks at its prey, then analyze the ways, and after some time attack. If we want to study cancer, there are probably different models, but we can use this one, blinded mole, that is known to not develop the cancer. So some scientists study it and hope that this method to combat cancer can be also applied in humans. But there are also other model organisms that we can use for experiments in the lab. So this is very important that these models are adjusted for the lab conditions and they are easy to handle. In case of animals like multicellular, here we think mainly about mouse, musculus, mouse, zebrafish, Drosophila, and of course Caenorhabditis elegans. So what is C. elegans? It is just 2 millimeter long nematode. Some people would think it's nothing special. Even the scientist who introduced C. elegans in the lab at the beginning was first interested in other species of the nematode. But it was so easy to keep it in the lab that at the end we end up having C. elegans in the lab. As you can see on the graph on the right, it don't take long to get adult from embryo. It is around 48 hours. And when we combine it with fast reproduction, around 300 embryos in 3 days, we can have a lot of words for our experiment in a very short period of time. Next interesting thing is that C. elegans' exact number of the cells, somatic cells, 959, and all of the worms are exactly the same in this matter. You may wonder why we have this photo on the right, photo of the people. It is photo from one of the C. elegans meeting, and quite often during these meetings there is more scientists than C. elegans cells itself. It underlines how popular this model is. Other important thing is to study C. elegans is that it is transparent, so we can see whatever happens inside of the worm. So here you can see several embryos and also oocytes on the down side of the pictures, and it's clearly see how many cells we have, nuclei. Some of the cells here are dividing, and even for the person who is not super biologist can recognize them. So it makes things easy to study in C. elegans. But that'll be simply nice model. What is most important is that this homology between humans and C. elegans. Around 50% of the human genes have orthologous in C. elegans. What was most important, around 2/3 of human disease-associated genes have also orthologous in C. elegans. So we can use this to study diseases, and it's what it makes it all combine very interesting model in the labs. So next step, why this even more easy? Here on the left, I'm showing you usual setup in the lab, C. elegans lab that we are using. It is enough to have just a flexion microscope. You can see a lot of agar plates with bacteria. We grow on them C. elegans, and this yellow marked is picker to move worms in between the plates. together with incubator 20 degrees. This is enough to have like main setup to C. elegans lab. But in many labs, we have many different strains with many mutations. What to do with them? What is nice in C. elegans, we can freeze them and keep them for years. My personal record was to take from the freezer a vial that was kept there for 25 years. And the student at the time I was working with was even younger than the worms we used for his experiment. To go even level up to this, we can send worms to strain library, and many scientists does it, especially with very interesting strains. So sometimes you don't have to even generate your own mutation and other stuff like that. You simply can order it for a relatively small price because it's 10 euro per strains. So then you can go for really broad scale in your experiments. So what kind of experiments we can at the end do with C. elegans? Mainly people expose C. elegans to different toxins, different drugs, different stress, or they see what happens if we have a different mutation in C. elegans. I cannot explain you and show you all possible experiments with C. elegans. It's probably hundreds of them. Probably the only limit is imagination of the scientists. Here, like typical, you could see almost in all labs. You can check effect of this, let's say, stress on number of the progeny, because of course it is very stable in C. elegans around 300 worms. You can see check time of the development, because when we start from the same point, they should reach adulthood almost at the same time. And they're even stable to the moment, to the way that if we keep them in a droplet of the water, they bend at the same ratio, the same worms. So this is relatively stable. But of course, people are doing even more fancy stuff with the C. elegans. Here we are mainly thinking of something cell biology, where we can study divisions, what happened with the organelles during different stages of development, and stresses and drugs, and also whole organs. But we can also study C. elegans at the level of the population, how they interact with each other. So it's not surprising that a lot of discoveries were made with use of C. elegans. I cannot show you all of them. I can assure you that many important pathways of proteins were first identified and described in C. elegans, but I will show you only these three because scientists doing this research receive Nobel Prize. So we can also say somehow that C. elegans is kind of champion in science. First of all comes from the early studies on C. elegans, where scientists discovered that cell death is kind of similar between different organisms, but they also studied organ development. In case of the second one, they had a quite tedious work to do. They traced development of each cell during development. That means from the one cell embryo to the adult worm that have 95 and 959 cells. Each of the division was marked and well studied, and we know what will happen from each cell at each point. Here on the right, you can see two embryos, different stages, 2-cell and 4-stage. But what can you also see here? There are granules called germ granules. On the left, they are on one of the 2 cells, and on the right, they are still on the one of the 4 cells. These granules go only to the cell that in the future will develop in the germline. And kind of similar process happened with all of the cells, so we will know what will happen exact cell from each of the division, so we'll make it very easy to study C. elegans. Another thing, another important discovery was RNAi interference. It is kind of way of repression of the expression. So how it works? It works like this. During transcription, we are getting mRNA that in external translation should end up as a protein. But when mRNA binds with this mRNA, then this mRNA is degraded. So we end up without protein at the end. So why it is so important? Because we can adjust it for the lab. There are libraries to mRNA against all possible genes in C. elegans, and every lab has such library, almost every lab. Here on the right, you can see one of experiments like that. Simply worms growing on the bacteria that has RNAi against HSP90. You see they are visible smaller than even dead embryos. This method was also applied for many different species, allowing to do a lot of different and nice experiments during this time. The third one, last one so far, but maybe not. Maybe we'll get more Nobel Prize for C. elegans. It is GFP or green fluorescent protein. As I said, it's quite similar because it's also about how it can be applied in the lab. For several species studied here for GFP. As you can see in the upper photo, we have a C. elegans, but in the down photo, we have a signal from fluorescence. We can tag different protein. Here there is a protein that is involved in the germline, so we can see only signal from the germline. But this way we can also tag different organelle and different processes. We can have also different tags and see how all these different proteins can interact, and we can even combine it with previously mentioned RNAi to see effects on this interaction. So it makes quite big toolbox for C. elegans and other species. But where we can exactly use C. elegans? Of course, none of the model species is perfect. We can use it only in a case when the process or the disease has evolutionarily conserved proteins or pathway. And C. elegans is used mainly to study diseases like obesity, aging, cancer, Alzheimer's disease, but also Parkinson's disease. So here on the right, you have a model representing how to use the C. elegans. You see that there is a model with human tau overexpression. Tau is involved in Alzheimer's disease. But what I want to underline here is that you can use C. elegans in two directions of studies. First of all, we can study mechanism of disease, what kind of proteins are here interacting, how they are interacting, to dissect the disease even more. And second, when we know exactly how it works and we have a clear phenotype, we can use it to screen for drugs against these diseases. So during my career, I had the pleasure to work with two projects. One of the project was focused on development of drugs, and second, it's rather on mechanisms. So I will just briefly show you how we can use the C. elegans. So soon after my PhD, I've joined a group of Oskar Hemmingsson in Sweden. It might be surprising to you, but he's a surgeon, so sometimes between the operation he simply picks worms. And in his research, he is interested in cancer, and to be specific, one of the pathway, RAS pathway or ERK pathway, that is most commonly mutated pathway in the cancer. And when we talk about mutation here, we think about overexpression. So the simple idea is in clinics, let's use inhibitors of the pathway, shut it down, and then stop development of the cancer. And to be fair, it works pretty nice. There is a lot of inhibitors already used in clinics. However, there are several different problems connected with them. So there is great need to find new and better inhibitors. And here we use C. elegans. So what's the pathway? It is simply cascade of the proteins that's transduced to the cell signal. But the signal is involved in apoptosis, differentiation, and proliferation. So this is clear, obvious that malfunction in the pathway, or in most of the cases, overactivation in the pathway, can lead to the cancer. So how we can use a C. elegans in such case when C. elegans doesn't have a cancer? First of all, C. elegans also has this pathway. It is evolutionary conserved. Matter of fact, one of the proteins in the pathway was first discovered in a C. elegans. However, in C. elegans, the pathway play a role in different moment. It is in specification of the vulva. what is interesting is what happens when we have overactivation of the pathway in C. elegans. It results in so-called multivulva phenotype, where more of the cells start to think that they should become a vulva. In case of complete shutdown of the pathway, worm develop without vulva. So we have this pretty clear phenotype showing us activation state of the pathway. So it makes like perfect model to study the pathway and use it in drug development. So we used our worms for development of an assay. We used the wild type pathway. So the idea is that if worm grow without inhibitors, it will grow with vulvae. But if the inhibitors is there during specification of the vulva, the worm will grow without vulva, so-called vulvaless worm. We introduced to our assay mentioned before GFP, so now you can see how useful it is. In the head of the worms, the green signal, it comes from the pharynx. It allows us to identify number of the worms. And in the middle of the worm, the green dot comes from the vulvae. In down photo, you see worm that grow in presence of inhibitor tramatinib. It is already used in the clinics, and I can see it's lacking this signal. So this way we can detect inhibitors. We wanted to have it in automatic way, so we used microconfocal microscope. This way we can have photos from the bright field, but also from fluorescence. And then with use of software, we can analyze this photo and based on that get number of the worms and get number of the vulvae. And this way track and find MEK inhibitors or inhibitors of the pathway. So at the end, we use it to screen it against library of more than 20,000 compounds. The part of the project with C. elegans is done. The compounds are still under evaluation. Here I'm showing you only small part of the sub-library. We detected here a lot of already known MEK inhibitors, pathway inhibitors, underlining or showing that we are truly able to detect such inhibitors. So as I said to you, this project is the C. elegans part project is done, and next year it will be a preclinical evaluation of these found compounds. After this project, I joined group of Ombre Selin in France. She is mainly interested in proteostasis. So what is proteostasis? It is simply homeostasis of the proteins. So exact level of all proteins have to be kept in a balance from the biogenesis through folding, trafficking to degradation. Cells have a way to keep it this way. However, when they lose it, it is quite dangerous for cells. They form aggregates. And how dangerous are they? Probably name of this disease that are associated with forming of the aggregates says a lot. And I don't have to prove you how important it is to study proteostasis. So in my project, I am interested in very specific part of the proteostasis, information of the stress granules. These are formed in response to the stress when untranslated mRNA is stuck together with proteins in higher structures called stress granules. This way cells can survive this stress. However, we don't know exactly what kind of proteins are there and what is role to this. So we want to dissect it. Here on the right, you have a germline of C. elegans tagged with GFP, and we use marker GTBP1. This is marker of the stress granules. You see on the photos below that when we expose worms on the heat shock, there is a lot of FODC formed in the cytoplasm. These are stress granules. GTBP, it's been studied in relation to the Alzheimer's disease, for example. However, this is not main focus of my study. I'm rather interested how the germline reacts to the stress. Nevertheless, what I want to show you is how it is possible to study this kind of processes in C. elegans. So in our lab, we implemented so-called TurboID method. TurboID is enzyme that violate everything in its surrounding, every protein. Here in our approach, this TurboID is connected with anti-GFP nanobody that will bind to the protein that is tagged with GFP, in this case, GTBP1. So at the end, TurboID tags everything, also everything surround, that means GTBP, and hopefully interacting proteins with GTBP. But why biotin? Because it forms very strong bond with streptavidin. So if we put into lysate beads covered with streptavidin, we can simply fish out proteins that are butylated, that means interact with GTBP. You may think that it cannot interact this way. Here is the proof. We, of course, every time doing a staining to see that the butylation occurs in exactly the same place where our bait, that means GTBP, is. And as you can see here, there is a colocalization. One of the important things for us was to have butylation occurring only in the moment of the stress. So we developed media without biotin. As you can see completely on the right, this butylation, when there is way more signal, occurs only when we add biotin, and we add biotin in the moment of a heat shock. At this moment, we have our first preliminary results. We will validate them. I won't show you them here because still a lot of work to see them. What I wanted to find them and validate them. But what I want to show you here today is that even very complicated things can be very easily done in C. elegans. So I was thinking about what kind of conclusion for this we can have it, but I think there's one that C. elegans, if you have a good question fitting to the model, it can be like really nice model. There is a lot of tools that we can use, and probably in the future they'll be developed even more additional. So I hope there will be even more. I'm pretty sure, to be fair, there'll be even more discoveries based on research on C. elegans. So at the end, I simply wanted to thank first of all funding bodies that helped me to perform my research during all these years, but also colleagues and friends that helped me. And of course, I wanted to say thank you for listening to me, and I hope I convince you that C. elegans can be a good model. Thank you very much.
That would be all for today. We hope that the subject and form of lectures were interesting for you. Thank you for your attention and presence.